So the next speaker is Cristianne Rijcken from Crystal Delivery in Maastricht in the Netherlands, and she will report about CriPec, Nanomedicines, principles, preparation, evaluation, and early clinical translation. Go ahead, Cristianne.
Yes, so good morning, everybody, and thanks for the kind introduction. And indeed, the title of today is Early Clinical Translation, as last year the title was Towards Clinical Translation. I'm happy to introduce to you today not only the concept of CriPec, but also the early stage result of the actual phase 1 study with the lead candidate product that we are developing. So we are a company actually named by now Crystal Therapeutics, but the official statutory name is still Crystal Delivery. But Crystal Therapeutics is a company established slightly more than 5 years ago and located in Maastricht, where we're founded and also financed by venture capital. So CriPec is our proprietary platform that allows for the development of different types of nanomedicines with a clear focus on improving the therapeutic performance, whereby you could entrap a range of molecules of APIs in this nanoparticulate system. So not only small molecules, but also larger compounds as well as up to oligonucleotides, of which I'm pleased to show you some initial data as well today. Not only can we entrap different range of compounds in there, but can we also entrap or conjugate compounds to the surface of these nanoparticles, as I'll also show you. And what we do ourselves is we develop products in the oncology area, but together with partners we work also outside this oncology area. The company is managed by these 4 people, of which Joost Holthuis is the CEO at the moment. And very recently we are joined by Edwin Klumper, who is also in the audience today as our Chief Medical Officer. And actually now maybe it's time to dive into what CriPec actually is as the underlying platform. And it's a polymeric-based platform where we generate nanomedicines in a 2-step approach, a really universally applicable approach. Namely that we take the drug compound, we derivatize it with a linker, and then if we combine it with a polymer, we create a nanoparticulate system which is stable as both the drug linker as well as the polymers are all covalently linked to one each other. So in other words, this nanoparticle is one big macromolecule, and if required or preferred, there is a single step needed to conjugate a targeting ligand to the surface. Well, as you can imagine, and has been explained in the overall conference so far, this nanoparticulate system has a completely different behavior upon administration to the body as compared to the native API that is entrapped. But what is important is over time, the nanoparticle will release the native drug again, as well as it will disintegrate into smaller fragments, which will ultimately be cleared by the kidneys. And it does so as a result of chemical hydrolysis between the drug linker bond as well as also the polymers that are also covalently conjugated via biodegradable bonds. And as we can use a whole range of different linkers as well as different biodegradable bonds between the polymers, you can really tune the features of the release kinetics. So overall, the system has a high tunability, and I'll also show you some data that we can make these particles as small as 30 nanometers all the way up to at least 100. You could go higher, but that's not of our primary interest. As indicated, the tunability of the system is high in terms of release rate. Conjugation is easy, and the overall approach of generating these nanoparticles is generic, so it doesn't depend on the API that is to be entrapped, but you can apply it for different type of APIs. And not unimportantly, it's already successfully upscaled towards GMP conditions, and the process as such is very straightforward and not as complex as, for example, liposomes. So when generating a novel drug product, I think everybody of us takes into account the current understanding of the biological situation. That's what we also take into account in developing new products, whereby I think CriPec has a highly tunability and it allows for the rational design of novel drug products, where based on the local pathophysiological features, we can modify the nanoparticulate properties such as to enable the best outcome. For example, a good circulation half-life or a selective release over time, ultimately leading into a higher efficacy as well as a better safety profile. And of course, this is not only applicable in general, but also specifically in tumor targeting, where our primary focus is. So I'll show you some data where we have nanoparticles that have a good accumulation in primary as well as metastasis, but also local sustained release over time. So the CriPec platform as such has a potential broad applicability, but as indicated, we're working with CriPec docetaxel. That's currently in phase 1 evaluation, and we're aiming forward to go to phase 2A early next year. In parallel, we have the so-called CriPec active targeting platform that's broadly applicable. We developed it together with partners, as well as also the combination with CriPec oligos, where we are still in the early stage, but there we aim also to go forward with partners. as we already have some partnerships, which I unfortunately cannot disclose, but we do have some partnerships with Nexigen and PX Biosolutions. So maybe dive into some actual data rather than talking about the concept. So last year I was already capable of demonstrating to you these figures where the efficacy was monitored in a breast cancer model, that upon equimolar administration to this breast cancer model, equimolar as compared to Taxotere, I should mention, then you clearly see after one single injection a long-term suppression of the tumor growth. But because of an improved safety profile of CriPec, we were capable of giving a higher dose, and that ultimately leads, in case of even an established tumor, also one single injection was sufficient to have a full tumor eradication over time, as compared to Taxotere, which only temporarily suppresses the tumor growth. And as indicated in kind of the concept slide, we anticipate to have a higher tumor uptake of the nanoparticles, and that was indeed demonstrated that in case of equimolar comparison, we have a much higher accumulation after either 2 or 4 days. And of course, if you go for a different dosage, like a higher dosage in case of CriPec, then the accumulation is even significantly larger. There's one thing I'd like to point your attention to, is that on the y-axis, there is descripted total dose XL. So that's the combination of the dose XL still covalently linked in the nanoparticle, as well as the material already released. And of course, as you are aware, it's only the released material that can exert a biological effect. So in recent years, what we have done is done an additional study, so-called AUC study, to monitor actually in the tumor tissue over time the exposure of the tissue to total dose XL, but also the released material. And that is depicted here, where you can see that in time, so this is up to all the way to 3 weeks, that you can see that total dose XL levels are fairly good. So there's a long-term accumulation of the nanoparticles. But even more importantly, you see also that dose XL is continuously being released out of these nanoparticles. They've been continuously suppressing the tumor growth, and that's for us the supporting evidence of the tumor regression as we saw in the previous slide. Next to that, we also evaluated the efficacy in a prostate-resistant model, which is depicted here. We did that in collaboration with the Erasmus University. So they exposed the cells continuously to dose XL, and they're not responsive any longer to dose XL. But in our case, we were capable of getting really long-term suppression of the tumor growth, where in case of Taxotere, the animals had to be sacrificed because of too much tumor growth. And then something that I also showed last year is about the PK profile. That's the differentiating part as compared to Taxotere. Taxotere rather rapidly eliminated out of the bloodstream in case you administered intravenously to rats. But in case of CriPec XL administration to rats, you see really long-term circulation of the open sphere representative for the dose XL, so the intact nanoparticles. And you also see release of the native dose XL, and even the metabolite can be measured. And in this case, in the rats, the half-life of the nanoparticles is approximately 70 hours. So this is what we had in mind, and then we did the overall, clinical development plan, applied for clinical trial. And I'm pleased to share with you the first data of it, namely the phase 1 dose evaluation, dose escalation study that is performed currently in Leuven and Rotterdam, whereby, in this case, the starting dose of the first cohort was 50 milligrams per square meter. And as you can see on the left side, this is representative again of the intact nanoparticles. Also in the human situation, we really have a long-term circulation of the nanoparticles, whereby, and I would have to point out your attention to the fact that this is a much lower value than in the case of the total dose XL. We can also measure the released dose XL in the blood of these patients. By now we have treated approximately a dozen of patients, and in this table you can see the PK parameters of the first 3 cohorts. Each cohort consists of 3 patients. And as you can see, the half-life of the nanoparticles is approximately 30 hours, so really long circulating, equal as to what was observed in the rats, though because of allometric scaling, of course, longer. And that's what you can see is that the dose corresponding parameters do increase. For example, the Cmax as well as the AUC is increasing with the dose, but the half-life remains equal. Of course, we are aware we're not the only one generating those XL nanoparticles, so we also took a comparison to the other parties across the globe who are developing. And I think Bind as well as Cerulean are the main, I would say, competitors. And as you can see, our half-life is, as compared to Bind, significantly larger. Hopefully, they're also resulting in much more enhanced tumor accumulation. And actually, the same holds true for Cerulean, who unfortunately did not yet present their half-life. But at least based on the AUC, we expect a much longer residence time in the blood and accordingly also much higher tumor accumulation over time. The phase 1 study as such is still ongoing. Safety parameters are being evaluated at the moment, but I cannot yet disclose these results with you. But maybe that's something for next year. So then I'd like to briefly touch upon the active targeting development as well as also the oligo, where in case of the active targeting, we took RGD as the model compound to be conjugated to the surface. And we did that via a so-called click chemistry approach, where on the surface of the nanoparticle we present an azide moiety. Of course, this is not to scale, but it's just to illustrate that if you conjugate the azide to cyclooctane moiety, you generate this triazole bond. And indeed, that was we were capable of monitoring this via 15 nitrogen NMR, really illustrating the generation of this bond, and it allows ultimately also to directly quantify the numbers of RGD molecules to be conjugated to the surface. And in combination with SLS, we figured out that we can really tailor the amount of ligands that are conjugated to the nanoparticle. So really the tunability is demonstrated here. Generated nanoparticles with RGD on the surface as well as doxorubicin in the nanoparticles. Well, as you can see, a small particle size with a low polar dispersity. And we tested these together with Twan Lammers in Aachen in an either integrin negative cell line or an integrin positive cell line. So RGD has an interaction with the integrin receptor. And as you may see, is that in case of the integrin receptor absence, you hardly see any difference upon increasing values of RGD on the surface. However, in case of the presence of the integrin, you see by increasing the percentage of RGD in the surface, you see an increasing interaction. So this is kind of a binding study, but if you even go forward and taking confocal studies with colocalization with a lyso tracker, then you see even further that there is an anisotosis of the CriPec nanoparticles and that the percentage is increasing with increasing percentage of RGD. So this is for us the initial proof of concept of the binding and the enhanced internalization. Irrespective of that, we also took a study in pharmacokinetic profiling whereby 35 nanometer sized nanoparticles were evaluated as compared to 65 nanometer sized with and without the RGD ligand. And I could have made an overlay of these 4 plots, but as you may see, the lines will be perfectly matching and falling over each other, indicating that the particle size nor the presence of RGD has an effect on the PK profile. And accordingly, we can say that the PK profile is not affected by the ligand on the surface. So very briefly, maybe also mention to you the accumulation of CriPec nanoparticles in metastasis. So former data were shown to you on the primary tumor, but also in collaboration with Aachen, we did an accumulation study whereby the tumor cells themselves were demonstrating a red fluorescent protein. The nanoparticles were labeled with a near-infrared label, and that allows for non-invasive monitoring of the nanoparticles over time, as well as also the metastasis and actually tumor formation. And you saw in time nicely the accumulation of the nanoparticles also in metastasis next to the primary tumor. And if you took out the animals, the organs out of the animals, you literally could see a nice colocalization of the nanoparticles in the primary tumor, but also in the different metastasis on the different organs. So that's really representative that these small size nanoparticles do have a good accumulation. And we're definitely going forward in evaluation also the efficacy in terms of metastasis treatment. I see the time is ticking, but I'd like to very briefly illustrate the oligonucleotides development so far, where we've generated pharmaceutical proof of concept at least, namely that we took along double-stranded siRNA targeted towards the AHA1 gene. And what we actually did is we took along the 5 amino prime double-strand siRNA, we derivatized with this linker with the different functionalities that allows for the temporarily entrapment within the nanoparticles. And then, well, we can use a range of linkers, but this is what we just focused on using the proof of concept. We generated nanoparticles out of it, and then this is kind of the control. So this is without the oligonucleotide, and you see even upon entrapment of different percentages of the oligonucleotide, we get an equal size, approximately 55 nanometers with a reasonable good polydispersity, and then an entrapment approximately of 60%. Whereby I also like to point out that the charge is masked by entrapment of the oligo in the nanoparticles. And then, of course, it's about, well, you can get it in there, but what about the properties of getting the oligo out of the nanoparticles? And then this depicted here. Depending on the different percentages, you could correct for it in terms of the release of the oligo over time. And that's what you can see is we have a very nice sustained release of the oligo from the nanoparticles over time without any burst release. And the confirmation has been obtained that the oligo that is released is really the native compound out of the nanoparticle. So the entrapment and the subsequent release did not affect it. So we think that based on this approach, we will also have a much more beneficial PK profile, as we do have an initial stable system. So we think we will not only target the liver, but also other organs. And actually, we're going forward for tumor targeting as a first preclinical proof of concept. So, make a long story short, I think CriPec is a broadly applicable platform with the first compound now in clinical development, and maybe next year I can show you some additional data in terms of the safety as well as potential efficacy data so far. And I'm more than pleased to answer any questions. So thank you for your attention.
So thank you very much for this comprehensive overview. We have time for one very short question. Yes.
Cristianne, thanks for an interesting update of what you're doing. The clinical work, what is the particle size that you use, and why did you choose the one that you have chosen?
We chose the 65 nanometer size, and actually thereafter we developed further the platform in terms of that we also went to smaller size. But I think 65 is, as compared to the majority of the liposomes, already smaller, and 65 should be having a good profile also in terms of sufficient drug entrapment in there.
But when you go to the smaller ones, is the drug entrapment the same?
It's slightly lower sometimes. Depends on slightly on the API.
In principle, that would be the preferred route to go, isn't it? The smaller ones, the more penetration in target sites.
Right. On the long run, the smaller the better, although there may be also an optimum situation. But it's kind of continuous development as it goes.
Okay. Thanks again.